All right, so a couple of people asked about the homework. So the other thing is, I got this nifty new thing. That's not me. Connect sent me a message and tattled on a few of you guys. Some of you guys have not paid for your Connect. You need to do that because obviously then you can't do your homework, right? So remember how I said before, if you absolutely have to, you can go stand on the street corner with a little sign that says, I need money for textbooks, and if I see you, I will give you $1. Okay? No excuses. Find a way. Anyway, beg if you have to. All right? This is important. Your education is important. Skip going to the movies this weekend or something. Find a way to afford it. Okay? That having been said, I put deadlines on homeworks, right? But I haven't been evil yet, and please don't make me have to be. I put unlimited on them and student view. And it'll tell me that you did it late, but I'm not going to take points off for that. Don't make me have to be that teacher. Please try and get it done at least once before the due date. The reason why I do unlimited, so you can do it again to practice, right? And tonight, yeah, today, I'm in pain. I'm doing the best I can to manage the pain. So, if I say something wrong, just correct me. Because I can only take one Tylenol, because if I take two, I'm out. Yeah, I think I might be calling the doctor after this and get it checked out. But I'm pretty sure it's just a pulled muscle, but it hurts like hell. Chapter 2, which I don't know why it's highlighted like that. Oh, I didn't put it until due tonight at midnight, so it's still open, active. I guess that's what that color means. I don't know, I'm used to seeing your guys' view. But if you haven't done it, just do it. But we're going to go ahead and start off class today because we had some questions about it by doing it together. And oh boy, it's small, huh? But you know what? You can blow things up, right? I'm in Firefox, and Firefox you go to the open menu, and you can hit over here, but apparently there's some other keys too that you can, quick hotkeys, you can blow things up, but my blind ass will just use the browser. Okay. And double click again. Okay, so what was this one? Okay, so you could use the animation. It says, 2 phenotypically tall heterozygous pea plants are crossed to one another. Okay, so that's what we got to start out with. So which one is phenotypically tall but heterozygous genotype?
Big T, little t.
Big T, little t. So we drag those 2 into the box. Okay? Did I show you guys how to use this? I thought I did. Okay. If I didn't, that's okay. All right. So then it says, a phenotypically tall F1 prodigy plant is randomly chosen and mated to a phenotypically dwarf plant. Oh. If some of the F2 generation offspring from this mating are phenotypically dwarf, what is the genotype of the, okay. So actually, they're telling you what the first generation is. Okay, so let's step by step through this. Okay. So let's just give me some blank thingies. I put some thingies in here. Here they are. Let's do a copy. Where's the copy? There it is. Okay. And we'll do a new blank PowerPoint And we'll call it homework chapter 2, right? And connect. And we'll paste this new one in. And we'll just delete what's in here. Don't drag, delete. And I'll just paste a whole bunch of these in case we need them. Okay. So, they said, right, this is the very first cross, right? So when we fill this in, we get that genotype, this genotype, and this one. We get all three, right? But phenotypically, and let's just write it in here, this is tall, right? What is this? Tall. Is this big enough for you guys, or I need to make it bigger? I can make it bigger. Okay. All right. And this one is also tall, and this one is dwarf. And it does this to me sometimes. It autocorrects. Okay. So that's the first generation. Now they're saying the next generation, they randomly picked one of these talls. Like, you can't tell when you look at these guys what their genotype is. So which one you guys want to go with? How many want to go with homozygous dominant? Nobody? Okay, we're going with heterozygous. So we... Oh. It somehow got... It's okay. So the two are phenotypically tall heterozygous, right? And they're mated with each other. That's the first cross we did, which is this one. So then now they're saying, out of these offspring, you're going to pick a tall one. So there's two possibilities as far as genotype goes, right? We're going to pick the heterozygous, because nobody picked the other one. So it says phenotypically tall prodigy from that F1 is randomly chosen and mated with a phenotypically dwarf. So what's dwarf? How do we write that? Two little t's. So let's fill in our square. What are we going to end up with? So this guy is tall, right? This guy is irritating me. And it's going to do it again. Now turn that off. And tall. Okay. So, if some of the F2 offspring, which is what we're looking at in our squares, right, that we just filled in, this is what they're talking about. If some of the offspring from this mating are phenotypically dwarf, what is the genotype of their randomly accepted parent? Well, we guessed right? Because that tall parent had to be heterozygous in order for us to get dwarf children. Does everyone understand that? Because the dwarf has to have two little t's, and so that tall parent had to have a little t, a recessive, non-functional gene. Let's do the other cross just for ha-ha's sake, right? So what if we had picked the homozygous tall and crossed it with a dwarf? What are all the offspring?
Tall.
They're all tall, and they're all what?
Heterozygous.
Heterozygous tall. Everybody still with me? Don't be embarrassed to say no. So because of that, because of how this one would come out, right, that parent has got to be, as we said, heterozygous. Because this other option would give us all tall offspring. So that was tough, huh? I really made you guys think. This is good, though. Unlike math, struggling in genetics homework is good. Because we learn from our struggles, don't we? Even with math. Okay. So then the choices. So we would pick this one, right? And this would help you check your crosses, right? That's what this is. So you hit help and it puts the cross in for you, right? And you can fill these in. And notice when it has percentages, you got to do percentage, not fractions like some people like to write. Okay? I'm not going to worry so much about those. All right, let's work through the next one together. So heterozygous tall pea plant is allowed to self-fertilize. So that's basically the same step as our first one before, right? Two heterozygous mating or a heterozygous mating with itself. The resulted prodigy is 25% homozygous tall, 50% heterozygous tall, and 25% dwarf. And that's what we saw, right? If all of the F1 plants are allowed to self-fertilize, what percentage of the F2 prodigy are heterozygous tall? Oh boy, that's a lot of crosses, huh? Okay, that's 3 different crosses, right? So we got to do homozygous tall, heterozygous, and dwarfs. And then we got to answer the question, how many are heterozygous tall? Okay, so here's our heterozygous cross. We already did that one. So let's see. Paste. Let's change these into homozygous. This is easy, right? They're all what? Tall, right? All T's mating with all T's, all tall, homozygous tall, right? Every single one of these squares looks like this. So same thing with dwarf, right? If we made a dwarf with a dwarf, what are you going to get? Nothing but dwarf. And what are they asking for? The question is, out of all these boxes, what's the percentage that's tall heterozygous, right? Which would be big T, little t. So what would it be? We have to take all these in consideration, right? So some of us do better with fractions, right? Sometimes not, right? So we only have 2 right here. But it's out of all of these offspring. So 2 out of 12. So that means 2 divided by 12. What does that come out to be? 6%? Is it? 1/6. Which is equal to, oh God, calculator. I should know this one off the top of my head. 1.66666666. 6. And we'll just round up. And that's a percent. Let's make sure that's one of the choices. What did we do wrong?
Because see, I think they're letting themselves fertilize, and 50% of them are heterozygous. So you have to.
Oh, you had to take that into account.
Take the percentages of how many you have started up into consideration.
Wow, this one's a tough one, huh? 25%, 50%.
So you have to look at how many of the 50% heterozygous.
Oh yeah, I'm not going to ask you guys this complicated of a question.
I think it's 25%. Because half of them are heterozygous, and half of the heterozygous. Like
Yeah.
if they're heterozygous, then half of 50% is 25%.
Somebody got this right on the homework.
We have to do 4 squares because there's 2 heterozygous offspring.
Oh, so that's how we could fix this. Yep, you're right. So we would have to basically copy this one. And then now, when we take all that into consideration, good thinking caps, y'all. Except I can't move this one right now. So we would count 4 out of 16. Ha ha ha. And then that gives us 1/4, which is 25%. Yay. I won't ask you that one that complicated on the test. I'll promise you right now. We save the complicated stuff for homework. It's when we finish, and that's a good question. So, I don't know if you guys noticed, I finally did at least part of our syllabus. So, let me open up a new tab. I added it under the syllabus module for us. And I had said this in class, but now I put it in writing for you guys, and I'll figure out how to get Canvas to do. I know Canvas can do what I want it to do. I just haven't done it in a long time. But you see under the syllabus and stuff, it says modules for our course. So after this chapter, we'll move on to chapter 4 and then 5, and then we'll have a test on that. So it's going to be a couple of weeks probably. And then test 2, module 2 will be chapter 8, 9, section 3 of 10, and chapter 11. And even some of these we might not cover the whole thing. Module 3 will be 12, 13, 15, and section 3 of 19. And then, remember I said I'm going to have you guys do some projects? And I'll give you guys details on that next week. And you can choose between these topics for your project. And you can choose for bonus to present this topic at the Science Expo. Or, sorry, STEAM at Delgado Day. STEAM Day at Delgado. Grading. So all the Connect assignments, any Canvas assignments like the discussion board introduction, which I haven't graded yet and you haven't done it, please do it. And your projects will make up 25% of your overall grade. Your exams, the 3 exams will make up 50%, and the final exam by departmental mandate is 25% of your overall grade, and it is comprehensive. Okay. So let's see, we figured out 25. Let's see what happens when I click check my work. Oh, it tells us we got it right. Cool. All right, did it open up another window? It did. Okay. So that was the first 2 questions. We got a couple more. Here's the arrow. Okay, so the next one, you can use this to help you, right? This is just to help. You don't have to do it. Okay. So when a heterozygous plant with round seeds is mated to a plant with wrinkle seeds, what fraction of the progeny are heterozygous? Okay, so let's fill in our cross. So round is dominant. Did I hit the wrong one? I did. Oh, it doesn't matter. Okay. So heterozygous, right? So big R, little r with a wrinkled, which is homozygous recessive. Am I right?
Yes.
Okay. Remember, in pain. You gotta help me out today. Fill in our thing so we end up with round and wrinkled. And round and wrinkled. And it's going to do that to me. Okay. And the question asks, I opened the wrong one. What fraction of the prodigy are heterozygous?
Half.
Tyler says half. When we look at our Punnett square, if I could open the right PowerPoint, I agree. Do you agree? Yay. That's the type of question I would ask on the test. The not so crazy one. Okay. Is that it for this one? Or is there a 4? There's a 4. Okay. When a heterozygous plant with round seeds is mated with wrinkle seeds, we just answered, homozygous. So this is the other half, right? Yes, yes?
Yes.
Okay. All right. 5. 2 heterozygous round plants are mated. What fraction of the prodigy with round seeds are genotypically heterozygous? At this point, we should know this one. 2 heterozygous give 1 homozygous dominant, 2 heterozygous dominant, and 1 recessive, right? So the answer is half. Yep. 2/4 is the same thing as a half. What? 2 headers.
Isn't it 3/4?
Let's go back. Heterozygous. Heterozygous would be black. Now I'm like second guessing myself. That's the cross, right?
It's because it asks what fraction of the ones with round seeds. So there's 3 with round seeds, right?
Oh, yeah. You got to read carefully. It's 2/3. Because they're just asking about the round ones. Not all the offspring. They were specific and evil. You got to read carefully. And I thought I picked the easy questions. All right, we should be right now.
That's 3/4.
Oh, what'd I do? Oh, I still didn't. Oh, let me change it. That's all right. We did okay on that one. What was the half? So this is where today's, let's see. Yay, see? Okay. If we get to today's stuff, reading carefully is very, very important. All right, so this was, we start getting into the dihybrid crosses. Do I want to torture us with this? Can we save these for Thursday? Give you guys a chance to work on them some more? Okay, let's do that. Okay, what are the last one? Oh, I made my thing too big and it's go back to center, Erica. Oh yeah, so basically this is, you know, find the 7 that he studied, right? You got to know the 7. It's one of your objectives. Stupid memorization question. Yep, we'll look at these next time. Okay, so do them if you haven't done them. We did the first couple. Help you out. Get back into the swing of things. Do you guys want me to post this PowerPoint? I'm going to save it though so I can reuse it. I'm going to do my homework before class. I'm going to be a good student too. All right, so back to chapter 2. And I think we were just starting dihybrids anyways. Did we get through dihybrid?
I think so, yes.
Yeah, and where you get that 9-3-3-1 ratio for 2 heterozygous. What we didn't do is, yeah, we went over that example, and then the big one. We didn't do fork line, right? So read over that for homework. It's actually in the part of your book here. I'm going to show you. So when you go to the book, and these are great, and I actually updated my PowerPoint, so I'll update it for you guys with some of the sample problems. So chapter 2, when you go to the ending of the chapter, it has the key terms and it has the summary, but then it has problem sets, right? And especially in your physical book, you'd have to flip to the back, and I think it's, oh, this one, they give you the answers for these. So here's that fork line. And you could even do this for dihybrid. So read over this, and we're going to go over it next time. I'm going to put together some PowerPoint slides for this. So this sample 3 on page 39 in the actual text, right? Read through this. And maybe even do that before you do your dihybrid crosses, because you could do your dihybrid using the fork line. And that might be easier for you than that big, huge 16 square punnet. I know I prefer to do that. So read through that first. See if you can figure that out. And we'll go over it first thing next time before we go over dihybrids. So then that brings us to modern genetics. Usually we're interested in the relationship between the molecular expression of the genes and the outcome of the traits. So the approach that we use is to identify an individual with a defective copy of a gene, which means it doesn't make a functional product, and observe how that affects the phenotype. And the interesting thing about Mendel, like I said, is he got lucky a couple of times over, and that all those that he looked at were actually this case where the recessive was a non-functioning gene. It didn't make anything. So the reason why the flowers are white is because they don't make the purple pigment. Okay? The reason why they're not round is they don't make the cytoskeleton structures, you know, the structural components to make it round, so it ends up wrinkled. It can't support that round. So we refer to this as loss of function alleles. And for us, as humans, a lot of times, a lot of our diseases are just that. We can't do something. We've lost that function. We've lost that ability. The gene we have is defective. That copy of the gene is defective, right? And unfortunately, both our copies are defective from both our parents. So as I said, unknowingly, every single of those 7, right? Round, tall, yellow, green, different pods, construction, right? Axial arrangement versus terminal. Probably need most of them. You should start learning them. Commonly, these are in a recessive manner, which means that you don't see that someone can't do something unless both their copies are defective. The other good copy is dominant over the recessive loss of function. So a pea plant has the genotype r, right? So that's recessive, right? They can't do something. In this case, they can't be round. They're what? Wrinkled. Big Y, little y refers to color, right? They're yellow. So how many different types of gametes can we make, and in what proportions? So remember that these are 2 different traits that we're looking at. This is basically setting up for a dihybrid cross. If you were going to set up your 16 square Punnett square, right? Or do your fork line. So little r, each one of the offspring is going to get one of these, but they're both the same, right? So all the offspring are going to get little r's, right? All the gametes that we're going to set up to give and make the offspring are going to be little r's. Everyone with me on that? Okay. And then they're either going to get a big Y or a little y. So if we were to write it out, right, so let's just say our first square would be little r, big Y. Another possibility is little r, little y. And then the other possibility, again, would be a repeat of that, right? Because everybody's getting little r's. So do you see that we wrote 2 of them twice? But again, you want it in lowest terms. So what we just wrote out, which one of these matches that? What are the 2 choices of gametes that we would make? B, right? Little r, big Y, or little r, little y. We have 2 of each, but 2 of 2 is the same as 1 of 1, right? Still with me? All right, so B's our answer. And this one, again, you got to set up, and I'm going to save this one for next time. But it's not 2 heterozygotes, right? So the answer is not 9331. We're going to leave that one for next time, as well as this one. Okay, so pedigrees. These used to be a nightmare for me. Right? But hopefully I will explain it in such a way that it'll make sense for you guys. So describe the features of a pedigree and analyze a pedigree to determine if a trait or disease is dominant or recessive. So when they draw out pedigrees, we just know phenotypes, right? We don't know genotypes. We're trying to figure out, potentially based on that family tree, what's going on for a particular trait or disease. All right. We can't tell people to mate with particular people to try and figure it out, right? We have to kind of back analyze. So we're going to look at pedigrees. So there are several symbols that they use in a pedigree. Circles are for females, squares are for males, diamonds are for unknown or not specified sex. Miscarriages would be denoted, I think, with just smaller symbols. Deceased individuals have a line through them. Unaffected individuals don't have their squares or circles colored in. Affected do. If they think that someone is heterozygous based on the evaluation of the pedigree, they will put a half shaded section. If they note that it's sex linked, it's a dot instead. Mating between related individuals has a double line between the two. And then fraternal, meaning they shared the womb at the same time, they have a little triangle that connects them. See how the slanted lines? And then they're connected. They have slanted lines for twins, but connected to denote identical twins. Because remember, these are genetically identical. And I did watch the link. Did I post it for you guys? So she was talking about that woman. She was, as I suggest, I figured it out. I was like, she's a chimera. And she was like, yeah, she's a chimera. What it was is that there were two babies that were fraternal, would have been fraternal twins in the womb at the same time. And they fused together to form one being. So that those mothers had two different sets of chromosomes distributed throughout their body. Two different sets. Because they were actually two individuals that merged as one. Really kind of freaky. Very rare. But it really posed a problem for them when they were doing genetic testing because they didn't match up with their babies. But when they did extensive genetic testing of them, they found out that they had two different sets of chromosomes on different organs of their bodies. Really kind of cool. So I'll post that link. It's a real short snippet from one of the news channels about that. And we may or may not, I don't know, I have to look what chapter it fell in, but we'll talk about chimera briefly. So that was really kind of cool. So here's a pedigree for cystic fibrosis. So again, the darkened squares are affected individuals. So they have this disease known as cystic fibrosis. These parents are presumed to be heterozygous because they're not affected, right? They don't have cystic fibrosis, but they ended up with a child with cystic fibrosis. This boy right here, number 3. So how did he have a disease that his parents don't have? Each one of the parents gave him a defective copy of that protein. And that's how he ended up with the disease. So each one of them are said to be what we call carriers when it comes to a disease like that. They're heterozygous. They are carrying a defective gene, but they don't have the disease because they have one good copy. And that's enough for this particular disease to be okay and not have the disease. So again, when you look at this side, you can figure out that one of his sister here is also heterozygous because she mated with someone else who also wasn't affected, but they ended up with children that have the disease, 2 of their children, which means that, again, both of these individuals are heterozygous.
So disease can't just get one generation in the family?
That's a complicated question that I don't feel comfortable answering right now. Can I come back to it?
Yeah.
Okay. I promise I will. Okay. All right. So this is a recessive pattern. And what really tells you that it's a recessive pattern is where you have unaffected parents, but their children have the disease. That's your clue there.
So on the left side where they're unaffected,
Yeah.
how do we know they're unaffected? Or are we just assuming because there's no children?
No. Okay, so this side,
Yeah.
so they don't have the disease. They mated, they have children that don't have the disease. So they may be homozygous dominant. Or this could be a problem of what? Do you guys know? Not enough data, sampling error, right? Maybe if they had more kids, they might end up with affected kids, and then that would tell you that they're both heterozygous. Or it could be as you see. And that's, again, the problem with looking at with pedigrees and not being able to control who mates with who, is that sometimes you need successive generations even to really kind of determine what's going on. But then again, nowadays, do we even rely on pedigrees? No, we just do genetic testing because it's cheap, quick, and easy. Right? This is kind of almost a thing of the past. So, it was commonly used, right, until genetic testing became very easy and cost-effective. So you have a normal allele, and usually you have a mutant allele, which is what we call the recessive, non-functioning loss of function, and that's usually what gives the disease symptoms. But that's not always the case. Right? If we have dominant and recessive, if you have a recessive pattern, which is what we just looked at, you can make two important predictions. Two normal heterozygous individuals will have, on average, 20% of their offspring affected, right? Two affected individuals will produce 100% affected offspring. Because that's all they have to give is defective alleles, right? So like we said with the dwarfs, all you're going to end up with is dwarfs. If you have cystic fibrosis and your mate has cystic fibrosis, would you probably choose to have children? No, because all your kids are going to have cystic fibrosis. If you know anybody with this, it's a fun disease. Yeah, and that's the other thing. For some things, sometimes we don't know because some diseases are so bad that people don't even make it to the age that they would actually procreate. So we can't even analyze their pedigree. Now, if you have a dominant pattern of inheritance, which means if you have that dominant allele, you have the disease. An affected individual will inherit the gene from at least one affected parent. So again, you only have to have one copy in this case to have this disease. Not cystic fibrosis, some other disease. And shame on them because they didn't give me an example in the book. And I can't think of one off the top of my head. That's too much. Alternatively, the disease may have been the result of a new mutation. So that's another thing too. You might end up with individuals that have a disease because there was a mutation that happened when the gametes formed, and that got passed on. They might not even have inherited from the parents, per se. So, cystic fibrosis, as I said, is a recessive disorder. 30% of Caucasians are carriers, which means they have one defective allele, but they have one good one, so they don't have the disease, but when they mate, right, they have a chance of having kids with this disease. And what it is a protein that's in your membranes called transmembrane conductase regulator. And what it regulates across the membrane is ions. So especially for certain cells and certain organs, this is a real problem. where they can't regulate ions across the membrane properly because this protein that they produce doesn't work. If they even produce a protein at all. I'm not even sure on that. So it leads to abnormalities in the pancreas and the intestines, sweat glands. The lungs tends to be the most encompassing and detrimental to individuals because, you know, breathing is kind of important. Just a little bit.
It makes everything really thick, right? Like really area.
Yeah, for the lungs it makes it really thick. Yeah, and again, it's a whole balance of ions against the across the membrane problem. So which of the following would not be observed in a pedigree if a genetic disorder is inherited in a recessive manner? So the same as cystic fibrosis. 2 unaffected parents, and the best way you can do is treat each one of these as a true and false statement, right? So 2 unaffected parents have affected offspring. Could this happen? Yes, and that's basically how we know it's recessive, right? Is that you have unaffected parents and then all of a sudden you have kids that have the disease. 2 affected parents have unaffected offspring. No, like we said, if you have it, you're going to give it to all your kids if it's recessive. So that one is the one that's not. So 1 affected and 1 unaffected parent have unaffected offspring. Could that happen? Sure. All of the above are possible? No, because we've already ruled out that 2 affected parents must have all infected children if it's a recessive manner of inheritance. That's all you have to give your kids. All you're going to give them is defective, right? They're going to have cystic fibrosis. So this is the questions at the back of the chapter. So this is number 13A, and it says to analyze this pedigree. All right, so let's start at the top. So we have this father who has it, right? He's colored in. This mom that doesn't have the disease. They end up with a child that has the disease, but the child does not have any children, right? So we'll skip over here. Their other children who don't have the disease and don't mate with anyone who has the disease, one of them doesn't end up with any children that have disease. The other one ends up with children that have the disease.
They must be heterozygous then, right? They both
Who?
They both must be, right? Her and the partner to be able to have.
Right, so these guys would have to be heterozygous, which means that's how these guys ended up with it. So this is a recessive pattern of inheritance, right? Where you, because you can see here that you have unaffected having, and it makes sense since they had an affected parent that this one could be heterozygous, right? Yeah, apparently so. Yeah. Yeah. Not a good idea. Okay. So, and again, so the answer is it behaves like a recessive trait, and the clue here was right here, right? Where a recessive trait, unaffected parents ended up with affected offspring. Now for the second part, do we have any uninfected parents giving affected offspring? No. But we've got infected parent giving infected kids every time. This is dominant. Did y'all see that? No, because just because you're affected doesn't mean you're homozygous, right? So they could be heterozygous, so they could be giving a good copy or a bad copy. The bad news is if they give the bad copy, the kid gets the disease. Make sense?
Like they're affected the father in this one. Does that mean the mother is hetero? Even though he's affected?
No, because it's dominant. all you got to have is one allele. So it's like being tall, right? All you got to have is one big T. So he is probably heterozygous, because if he was homozygous, all the kids would have it. Because that's all he has to give, is that defective gene, which is a dominant gene in this case. So the first thing I always do when I analyze these is to see, do I have any parents without the disease having children that have the disease? That's a clue that it's recessive. If that isn't the case, then you look and you see, like you do in this one, that if a parent has it, they always end up with kids who have it. But again, this could be a sampling error, because remember, it's a 50-50 chance as to whether they get it or not, if that individual is heterozygous. So as it said, these can be confusing, right? So you're really glad none of them will be on the test, right? But I will ask you the two clues, right? How do you know it's recessive? Because you could have two unaffected parents have affected offspring. How do you know it's dominant? Where's my list? It's in here somewhere. Here we go. Right? At least one of their parents is going to be affected. If they have it. If it wasn't a new mutation. So this is your key right here. Right? And the fact that if they have it, if it's recessive, if both parents have it, 100% of the kids are going to have it. Okay, so probability. What's the probability that I'm going to start probability? We'll start it. We won't get through it all. Okay. So we can't determine for sure, right? But definitely we can counsel people, especially when it comes to cystic fibrosis. We know it's recessive inherited. We know if both parents have it, all the kids are going to have it, right? We would inform individuals of that. But in other cases, right, especially like with sickle cell, you end up two parents that don't have sickle cell, right? And then they end up with kids with sickle cell. We can explain now why that happens, right? Because again, it's recessive. Both parents were carriers for a defective trait, right? And that's also a thing to take into consideration if you have sickle cell. That's all you have to give your children, right? Is that trait. So they can help them predict the likelihood, right? Make educated decisions on whether you would want to have children or not. So the probability of an event is the chance that it will occur in the future, right? We can't guarantee under certain circumstances, especially with heterozygous individuals, that your kid will have it, but you have about 25% chance. So of course we can mathematically calculate this, right? The number of times an event occurs over the total number of events. So flipping a coin is the easiest one, right? You have two choices, heads or tails. So the probability will be heads over the number of events, heads plus tails, is 1/2 the time or 50% of the time. So if we were to flip coins, and I didn't bring coins in today, mostly because I didn't want to hear them. What's the likelihood that half the time you're going to get heads? 50%. If we were to actually do it, say we flipped a coin 10 times, how many of you guys think you would get heads 5 times? Nobody? Why not? Too small of a sample size. Right? And that's the problem too with pedigrees and people mating and stuff like that, right? It's a really small sample size. You're more likely to get the expected, actually observe the expected, the larger the sample size. So when we deal with really small samples, like only doing it 10 times, and we say get it like 6 and 4, right? 6 heads and 4 tails. That's referred to as random sampling error. And so this error can be very large, right, if you've got a really small sample. So it's better if you have a larger sample, your error will be much smaller. So 10 times, you might get something like 7 out of 3. However, if we did it 1,000 times, you're more likely to come up with the 50-50. And that's why, too, when you look at Mendel's crosses, I mean, he didn't do just 10, he did like 1,000 crosses, right? And so that's why his numbers he was able to take those numbers and analyze them and kind of figure out what was going on with that pattern of inheritance. He was able to back calculate. So there are 2 ways that we can approach probability mathematically. One is referred to as the product rule, and one is referred to as the binomial expansion equation. And when you look at these, if you have a fear of math, it'll probably exacerbate that. The good news that I'm going to tell you guys in this class, we're not going to calculate out, this is not math class. Thank God, right? We're not going to calculate out these, right? I want you guys to be aware of them and how you would maybe calculate them out. What's more important is when would you use each one and why. So I'll give you homework problems. I'll probably have to do them outside of Connect. I'll probably have to maybe put them in Canvas. Because I'm just going to ask you the first half of the problem. I'm going to say, what would you use and why? Right? Why would you use product rule instead of binomial expansion? Or vice versa? Why are you choosing to use binomial expansion instead of the product rule? Does that make sense to you guys? So it's kind of like in physics, like which equation do you use, right? So it's the same thing. Which equation for the problem you're presented would you use? Which apparently we did turn our caps on really well today, earlier when we were reading some of those crosses, right? The same is definitely true here, and there's definitely some keywords that you need to look at. So the product rule is going to be used when the probability that 2 or more independent events will occur is equal to the product of their respective probabilities. Oh my God. So independent is important, right? That means that these 2 events are not linked to each other. Make sense? So each time we flip a coin, it's not affected by what happened before it, right? Each time we flip, whether we get heads or tails, is an independent event. So each time you have children is an independent event, right? What one kid had or doesn't have doesn't determine whether the next kid is going to have or not have. Does that make sense to you guys? Right? So if one kid has cystic fibrosis, it just means unfortunately in the lottery he lost. Right? He got 2 defective. The next kid is going to be his own set, right? Whatever randomly combined from both parents is going to determine what that child is. Everybody still with me? Okay. So consider congenital and I can never say this one. It's where you don't feel pain. It's a recessive trait in humans. They can't distinguish between sensations. However, extreme sensations are not perceived as painful. So 2 alleles, right? Normal gene, right? Denoted P. I don't know why they picked P. And congenital and yeah, whatever. Today is not going to come out of my mouth. Little p. Recessives are always little p. Question. 2 heterozygous individuals plan to start a family. We know this cross, don't we? Right? They're 2 heterozygous. The offspring. What's the probability the couple's first 3 children will all have congenital and whatever? Okay. So, this is a phenotype. We know this cross, though, right? This is your heterozygous versus heterozygous. Doesn't matter which Punnett square, right? Let's just look at a heterozygous one. So, it's this one right here. Just imagine they're P's instead. So, 3 of them are going to be normal. 2 are going to be heterozygous, though, right? One of them is going to be homozygous. Doesn't matter. Their phenotype is their normal, right? 1/4 or 25% will have the disease. Okay, so they say they're going to have how many kids? They're going to have 3 kids. What's the probability that the first kid is going to have the disease? 25%. The second? 25%. The third? 25%. But can we do this in percentages? No, this one has to be done in fractions. So it's 1/4 times 1/4 times 1/4. All right, so we already figured out it's 1/4, right? You could do out a Punnett square if you need to, or if you know your heterozygous cross. Then we're going to multiply. All right, so we get 1/64. Then you convert it into a percentage, right? 1 divided by 64 ends up being 0.016 times by 100 to give you a percentage, which would give you 1.6% of the time, right? Would a family under those conditions end up with 3 children with the disease? When you use binomial expansion equation, and this is the equation, right? You look at it, you're like, are you serious, Miss Erica? This represents all of the possibilities for a given set of ordered events. So then this is when we say, what's the likelihood, right, of the first, second, and third all having a disease? So it's a complicated equation, which by no means am I going to make you guys memorize or even calculate for that matter. But P is the probability, N is the number of events, X is the number of events in one category, little p is the individual probability of X, right, the number of events in the category, and Q is the individual probability of the other category. So of course, again, you can only do this when you have 2 choices. So P plus Q must equal 1. that symbol denotes factorial. That means that the number is times by all the progressive numbers before it. So if you have 4 factorial, it's 4 times 3 times 2 times 1. Again, we're not going to calculate this out. One exception is 0 factorial is actually 1. When would we use this? So when you have something like 2 heterozygous brown-eyed individuals have 5 children, and they want to know, what is the probability that 2 of the couple's 5 children will have blue eyes? So given that information, you could fill in that crazy equation, couldn't you? So you got to know your probability. Probability of blue eyes versus brown eyes. Blue eyes is recessive. So 1/4 and 3/4. Determine the number of events. How many events were in that? How many kids? How many kids? 5. The number with blue eyes is 2. We would fill it all in. Fun, fun, fun. No, no, no. Right? 26% of the time. Is it possible to have a kid with blue eyes? Yes.
So I'm saying, see, I've seen parents like both parents have blue eyes and they have 5 kids and I don't have the blue eyes. Is it possible to do that?
Yeah. So I hate that they use this one as an example. Because I think from what I remember, eye color isn't a single set of alleles. It's 2 or 3 in combination to determine eye color. So.
That's how hazel eyes come.
Yeah, like mine. Mine are hazel. It's on what color I wear. They look more green or blue. They're kind of both. It's referred to as hazel. And some people are more on the green spectrum of hazel or blue, but really it does also affect what's around you. The color that's around you as to what your eyes will appear to look like. If you have that mixture in your iris. And blue, again, is true blue, is no pigmentation at all. So if you true blue, it's 100% recessive. No color. Okay, so we'll save chi-squared for next time, obviously. But work some more on that same homework I gave you guys before. We're going to do some dihybrid fork line stuff, right? And then for the next homework, I'll give you probably in Canvas, because I think Connects is going to want you to calculate out, and I don't want you to calculate out. I want you to figure out what you're using and why.